Hello, my name is Lyla, Abbas.
My name is Feza, Abbas.
And my name is Matthew Hunt.
And today we will be discussing the development of Sharkavir, a new hypothetical inhibitor for HIV-1 protease. Our CREST team, which stands for Connecting Researchers, Educators, and Students, worked in conjunction with the Center for Biomolecular Modeling. As part of the CREST program, teams of undergraduates across the country worked to model the research of an Association of Biochemistry and Molecular Biology Award winner. This year, the researcher selected was Dr. Celia Schiffer. We chose to model her work on HIV-1 protease and its inhibitors due to HIV being one of the most dangerous global pandemics humanity faces today. Instead of showing our work at the ASBMB National Conference due to COVID-19, we have prepared a video of our presentation so that we can virtually share our work. The Center for Biomolecular Modeling, and particularly the CREST program, chose the work of this award-winning scientist, Dr. Celia Schiffer, for this year's modeling project due to her large collection of protein database files and her focus on designing antiviral drugs that are able to avoid resistance. She co-founded the Institute of Drug Resistance, and she's known for her work in researching drug resistance and understanding pandemic viruses such as HIV and hepatitis C. So, by designing drugs to be less susceptible to drug resistance, these drugs are more likely to last and stay effective. After studying the research from the Schiffer lab related to HIV-1 protease, we chose to focus on this key research paper that she worked on, which discusses mutations in the active site of HIV-1 protease. We chose to study the system due to it being a global pandemic, and we were particularly interested by one of the key components of the HIV life cycle, which is the enzyme HIV-1 protease. This enzyme is responsible for cutting up newly synthesized non-functional protein components for the HIV virion into smaller proteins that are able to infect other cells, and it does so by hydrolyzing peptide bonds. Currently, competitive inhibitors that belong to the class of aspartic proteases are used as treatment, but of course, due to the rapid mutation rate in the active site, this weakens the effect of these inhibitors. Here, we see that the HIV protein infects our immune cells, specifically CD4 cells, that are responsible for fighting off viruses. This is what makes it difficult to treat. However, with the help of protease inhibitors, we're able to halt the viral replication of HIV by inhibiting the key enzyme HIV-1 protease. The mechanism involved in preventing the viral replication is the use of protease inhibitors, which blocks new immature HIV from becoming mature HIV. This is the mechanism of action for aspartic proteases, including HIV-1 protease. This demonstrates the cleavage mechanism of the peptide bond through hydrolysis due to the two aspartic acid 25 residues that sit in the active site of HIV-1 protease. The highlighted hydroxyl group is part of the tetrahedral intermediate that mimics the transition state of the proteolytic cleavage mechanism. This is why one of the inhibitors used today, Darunavir, is such a strong inhibitor because it has this critical hydroxide that acts as a transition state analog. In doing research on effective protease inhibitors, we found that in certain cases, a mixed dosage of Darunavir and Ritonavir proved to be effective. Inspired by this clinical application, we decided to come up with a hybrid of these two inhibitors named Sharkavir. We named our hypothetical drug Sharkavir to honor our school's mascot being a shark. We can also see that critical hydroxide highlighted here. Our team's research question this year was to figure out how can molecular models be used to simulate the binding of various HIV-1 aspartic proteases, including Darunavir, Ritonavir, and a hypothetical combination of these two named Sharkavir.
As we move on to our methods, we started with these 3 protein database files to design the molecular models into JMOL. This allowed us to manipulate the crystal structures. The PDB files were 1T3R, this was a non-mutated HIV-1 protease that had Darunavir bound to it, as well as 40QF and 1N4. We also had our own PDB file for Sharkavir. Marvin Sketch, which is a chemical drawing software, allowed us to create the outline of our novel inhibitor, Sharkavir. Key amino acids that commonly mutate in HIV-1 protease were found from the literature, and we focused specifically on the aspartic clamp as part of the catalytic triad. This triad consisted of 3 important residues, aspartic acid 25, threonine 26, and glycine 27, as highlighted in orange. There were also 5 other key amino acids not part of this triad, but prone to mutating, and those were aspartic acid 30, isoleucine both at positions 84 and 50, glycine 48, and valine 82. Here are results and model information. First, the protease inhibitors were 3D printed using the spacefill option and light CPK colors. We can see here that a section of ritonavir and a section of darunavir was focused on, and Sharkavir, our hypothetical new drug, had aspects of both. The critical hydroxides that interact with that aspartic acid 25 residue as part of the catalytic triad are highlighted here with the green asterisk. Here we can see in JMOL a particular view of the active site binding box. The catalytic triad with aspartic acid 25, 3, 26, and glycine 27 is highlighted in orange at the center. Additionally, in green, we can see that there are 2 copies of each of the 5 key amino acids that commonly mutate since this is a homodimer. We then simulated how the HIV-1 protease active site would bind with each of the inhibitor molecules, darunavir on the left and ritonavir on the right. Both are colored in black for clarity. Then we used a different view using Autodoc Mina to show how our hypothetical drug Sharkavir would bind to the HIV-1 protease. The catalytic triad is highlighted in red. The next series of slides focuses in on our actual 3D printed model that you can hold in your hand that shows these key amino acid residues and how our substrates would be binding to the HIV-1 protease. In this view, we only see the active site binding box without any substrates added. Then here, we can see the HIV-1 protease binding box with darunavir bound into the active site. And note how that catalytic hydroxide is interacting with that aspartic clamp. Again here, we see the ritonavir substrate bound into the active site binding box, and we see that aspartic clamp interacting with that same hydroxide on the inhibitor. And lastly, when we were able to put in our Sharkavir model, we see that it also has that same common hydroxide interacting with that aspartic clamp.
In summary of our model information, we chose to model the 3 protease inhibitors, Darunavir, ritonavir, and Sharkavir, as well as an HIV-1 protease active site binding box. It consisted of 2 homodimers, as well as a catalytic triad consisting of amino acids, aspartic acid 25, threonine 26, and glycine 27, as well as some other commonly mutated amino acids. Sharkavir as well shown bound to HIV-1 protease, and as well the other 3D printed models inhibitors were able to be placed in the HIV-1 protease binding box. The development of Sharkavir is based on previously established inhibitor structures, Darunavir and Ritonavir. It can be used as a model towards the in silico computer-based development of future inhibitory molecules. Sharkavir still maintains the benzyl ring that's sensed by the specificity pocket, as well as the hydroxide that mimics the transition state intermediate. Using chemical hallmarks such as these present in known inhibitory molecules, we can make future combinatory drugs, as well as subsequently synthesize future drug testing. To further propel this project idea, we want to attempt to dock the novel inhibitor Sharkavir and see its effects as an inhibitor, whether it's weak or strong inhibitor. These molecular modeling techniques described are accessible to students and researchers and can be a great first step in the long process of drug design. We realized through working on this project that looking at these 3D models really helped visualize how HIV-1 protease works. The implications of these model inhibitors displayed how novel drugs can be conceptualized and serve as enzyme inhibitors for some of the world's deadliest diseases. This modeling project also allowed students to participate in drug design and introduce a research-based curriculum for classrooms. And here you can see a couple of photos of our CREST team, such as the photo on the right, which was taken on the day that our team received our protein model in the mail. Now, as a team, we would like to acknowledge a couple of groups and organizations that really helped us propel our project, such as Dr. Mark Franzen for her specific guidance on this project, as well as the entire team at the CBM for their encouragement in making 3D molecular modeling more accessible and cost-effective. The CREST program for supporting and making this project possible, Nova Southeastern University, NSU Pan-Australia Travel awarded to Feza Abbas, Lyla Abbas, and Matthew Hunt, as well as the ASBMB Travel Award awarded to Matthew Hunt, and Craig Lavin for photographing the molecular models. Now, this was one of our team's first online Zoom meetings due to the coronavirus and us practicing our social distancing. As well, if you guys would like to contact any of us, you can contact Dr. Arthur Sikora at asikora@nova.edu, as well as Dr. Emily Schmitt Lavin at eschmitt@nova.edu. Now here are all our references that helped us create this project.
Thank you for watching.